Hello guys, myself Archana Kumari and I can share some important notes about PCR. PCR. PCR stands for polymerase chain reaction. PCR is basically a technique of making multiple copies of a DNA fragment and it includes in vitro technique. History of PCR. In 1983, it was invented by Cadimulus. In 1985, first publication of PCR by Kitas Corporation appears in Science. In 1986, purified tech polymerase is first used in PCR. In 1988, Perkin Elmer introduces the automated thermal cycler. In 1989, science declares tech polymerase molecules of the year. And finally, in 1993, Cadimulus got Nobel Prize for PCR. Let's talk about purpose of PCR. To amplify a loads of double-stranded DNA molecules with same size and sequence by enzymatic method and cycling condition. And what is the condition of PCR? First, denaturation of double-stranded template. Second, annealing of primers. Third, extension of double-stranded DNA molecules. What is the steps of PCR? First, denaturation steps. It is the first step of PCR. At 92 to 94 degrees Celsius, the two strands of DNA they get separated. So double-stranded becomes single-stranded and each single-stranded DNA function as a template. Denaturation time depends upon two factors. Length of the DNA template, that is more length of the DNA has more denaturation time. Amount of GC content and AT content. You can see this diagram. Second, annealing. In this step, the temperature is usually 50 to 60 degrees Celsius. At that temperature, the primer allowed to bind to template DNA and its time duration 15 to 30 seconds. And the primer that binds to template DNA at the 3' end of the single-stranded DNA. You can see annealing steps. Third, extension. At 72 degrees Celsius, tech DNA polymerase attaches to one end of each primer and synthesizes new strand of DNA that is complementary to the template DNA molecules. Finally, in this step of PCR, now we have 4 strands of DNA instead of 2. You can see this is the steps of extension. What is the chemical components of PCR? Magnesium chloride, buffer, DNTPs, primers, DNA polymerase and target DNA. Basic requirement for PCR reaction is DNA template, primer, DNA polymerase, DNA nucleotides, double distilled water, PCR buffer. What is the types of PCR? You can see this is all the types of PCR and the application of PCR and another application of PCR. So thank you guys. Please like and share this channel.